Well, thank you very much for the invitation to join the symposium. Today I'm presenting an overview of recent work that I and my colleagues have undertaken aimed at understanding hominin locomotion from fossil footprints. And I will apologize in advance. I tried my best to translate my slides into French. It's been about 20 years since my last French class and the last time I wrote anything in French, so I apologize for any mistakes in my translation. So I don't have to convince anyone in this audience that locomotion is interesting or that it's integral to the way that any animal exists within and engages with their environment. It's one of the most fundamental reflections of natural selection. If we look at our closest living relatives, the apes, each species has a unique set of adaptations for moving around their environment in a particular way. Orangutans and gibbons climb trees in two very different ways. Gorillas climb to varying extents, but mostly knuckle walk on the ground. Bonobos and chimpanzees have their own unique ways of knuckle walking when they're on the ground and of climbing in trees as well. Us humans, on the other hand, we're very different. While there might be some similarities among the other apes that tend to use all four of their limbs for locomotion, we are the only ones who walk and run upright on two limbs instead. And our peculiar manner of bipedal locomotion is considered a defining feature of modern humans, and how we came to walk this way is one of the big questions around which our discipline is built. Now, this here is one representation of our hominin family tree as currently known. And every species depicted here is thought to have been bipedal in some way, shape, or form. But even though they all walked upright, there was almost certainly a great deal of diversity in how exactly each one of these species accomplished that behavior. I'm not going to venture into a full exploration of all of that diversity today. Instead, I want to focus just on this time period from about 2 to 1.5 million years ago. This is the time period in which I tend to focus most of my own research efforts, and I find this interval very interesting because it captures a mix of different hominin species, several from adaptive groups that are sort of fizzling out right around this time period, but one species from a group that's just emerging, and that species from a group that is just emerging is, of course, Homo erectus. Homo erectus is thought of as representing a really dramatic evolutionary step in the human-like direction. They're basically as strong of a candidate as currently exists for being a direct ancestor of ours. And of key importance is the fact that Homo erectus, from the neck down, looks quite similar to modern humans. If we look at this 1.5 million year old Homo erectus skeleton that was found near Nariakotomi in Kenya in 1984, we can see that it's very human-like in its overall morphology. Based on that human-like skeletal morphology and what we know about the ways in which skeletal form relates to function, people have hypothesized that this individual and other members of Homo erectus would have walked and ran in very similar ways to modern humans. Digging a little deeper into how we get from fossils to hypothesized locomotion, researchers like myself and many others in the room today who are interested in what we call functional morphology typically search for the ways in which functional signals can be inferred from skeletal anatomy. Before any inferences can be drawn from fossils, a great deal of experimental research is necessary to understand how exactly functional signals are reflected or recorded in the anatomies of our bones. So the examples here are just some of many experimental studies that have shown how things like joint morphology, trabecular bone anatomy, and the cross-sectional geometry of cortical bone reflect the capabilities of and/or the mechanical stresses experienced by these structures during their corresponding organisms' lives. So these studies form the mechanical, they dictate the mechanical connections between form and function, which paleoanthropologists can then use to start to understand from fossils how our extinct relatives moved around their worlds. The realities of the fossil record make life pretty difficult for us paleoanthropologists, though, because the fossils that we might hope to understand and interpret are very likely to be few and far between. On the screen right now are basically all of the foot fossils that we currently know of from Homo erectus. There's a small sample from Ethiopia and Kenya, and another small handful of bones from the site of Dmanisi in Georgia. Now, this is far from an ideal situation if we want to understand the full picture of what Homo erectus feet looked like and how they might have functioned. That reality is a large part of what has driven me to think about what other kinds of evidence might exist which we can use as alternative or complementary means to better understand the evolution of human locomotion. And one of the rapidly emerging lines of evidence that I've spent a lot of time focusing on is fossil footprints, which are essentially snapshots of locomotion itself. Within these snapshots are direct evidence of complete foot anatomy and the way that these individuals were walking at specific moments in time. And these pieces of information can be incredibly valuable then for understanding hominin locomotion at various points throughout human evolution. This kind of evidence hasn't been studied nearly as extensively as fossil bones, though, because for decades hominin footprints were extremely rare. The two sites on the screen here were both discovered in the late 1970s. On the left, the famous site of Laetoli preserves a set of 3.66 million-year-old footprints attributed to Australopithecus afarensis. The photo on the right shows a not nearly as extensive set of 1.4 million-year-old footprints found at Kubifora, Kenya, and we're not sure which species made these ones. For about 3 decades that followed these two discoveries in the late 70s, these were the only sets of early hominin footprints that were known that predated modern humans. Aside from one single isolated footprint that was found at the site of Terra Amata near Nice, which could be up to 400,000 years old, but there are some questions about those dates. But the quantity of evidence has really changed in the past 15 years or so. As I was entering graduate school in 2009, this discovery was announced of these 1.5 million-year-old hominin footprints found near Illoret, Kenya. At the time, I knew that I was interested in studying locomotion and the evolution of early Homo. They believed that these footprints might have been made by Homo erectus, so I was very excited to be able to join this project as an early PhD student. Now here we're looking at a Google Earth image of the fossil deposits in the Illoret area, and if you look at the scale bar down in the bottom right, that represents 200 meters, so we're looking at roughly 1 square kilometer here. Now that initial 2009 publication documented 2 footprint surfaces that were found at the same site, which is designated by the yellow pin here. Those 2 surfaces together preserved a total of 20 hominin footprints. From 2010 to 2012, I and my colleagues expanded that excavation. We nearly doubled it in size, and we ended up finding that there were a whole lot more. We increased the sample size at that site to a total of about 73 hominin footprints. Then in 2013, we also started looking in the surrounding area for more fossil footprint sites. And here, Kay Berensmeyer, who had discovered those Kubifora footprints back in the 70s, trained me and others on how to interpret geology and sedimentology to identify where these kinds of sites were likely to be preserved. Within just 2 field seasons, we quickly found that these kinds of sites were all over the place here. On this image, sites with hominin footprints are designated by the yellow pins, and sites with just the footprints of other animals are designated by the blue pins. So by 2014, we had found 25 different sites that together preserved more than 475 fossil footprints. Among these were 7 surfaces that preserved a total of over 100 fossil hominin footprints. Even more recently, our ongoing field research in the Turkana Basin has continued to be extremely productive. We're still finding lots of new and exciting things. This photo is from 2022, when I was working with colleagues here to excavate another footprint site at Kubifora that is just slightly older than any of the others we had found previously. After excavating this one, we uncovered another just spectacular set of hominin footprints. There is a very long 13-step trackway going from left to right across the screen here. There are 3 isolated footprints from 3 different hominin individuals who were walking in a roughly perpendicular direction. And there are other footprints from fun animals like massive storks and bovids and equids all around those. Now, we are still working actually to publish this site. We're at the final stages of that process right now, so I'm hoping that you'll hear more about it very soon, but unfortunately I can't share all of the details about it today. Anyways, in our new reality, after all of this field research, we are not starved for data anymore. We've uncovered 7 different sites and actually 9 distinct footprint surfaces at Illoret and Kubifora, all of which are between about 1.6 and 1.4 million years old. Across these sites, we've excavated 137 hominin footprints so far. There are certainly many more just sitting there waiting to be excavated at several of these sites. But that total sample size is already roughly 6 times more than the number of foot bones that have been recovered from this same general place and time. And people have been searching for fossil bones in the Turkana Basin for roughly a little bit more than 50 years now at this point. But we've only recently begun searching those same fossil deposits for fossil footprints. So I think I can say with a reasonable level of confidence that there are more discoveries yet to come here too. So I've just been focusing on sites where I've worked, but I also want to emphasize that within just the past 5 years, we've seen an amazing surge of footprint discoveries from around the globe. These sites span an impressive range of space and also time, going from about 3.66 million years ago all the way forward to about 10,000 years ago. And these sites are giving us an influx then of new data that are now available for understanding hominin locomotion at all of these different places and times over the past several million years. So finding these new fossils is exciting. We know that they're giving us amazing snapshots of our extinct relatives. But then we have to ask the question of how can we actually use these data if our goal is to better understand the evolution of human locomotion. And to do this, we have to approach this as a question of functional morphology. We want to understand how the morphology of footprints reflects or records the functional pattern or the locomotor pattern that produced them. Here, new technologies that weren't available until very recently have allowed us to actually lift up the hood and study exactly how a footprint is made. These two researchers, who are now close colleagues of mine, Peter Falkingham at Liverpool John Moores University and Steve Gatesy at Brown, had started just over a decade ago using biplanar X-ray and computer simulation to study the process of footprint formation in birds. They wanted to better understand the fossil footprints of bird-like dinosaurs. As they were developing these techniques, I reached out to them and we agreed it would be great to try to do this in humans too. And we've been able to do some pretty exciting things in the time since. So over the course of 9 years now, we've been developing and implementing biplanar X-ray methods that we can use to study how human footprints are made. To give you an idea of what our most recent experiments look like, we first capture a high-resolution 3D scan of someone's foot. The little black dots here indicate the positions of tiny radiopaque markers that we adhere to their foot. People then walk along a foam-covered trackway with a radiolucent mud at the center of it within the space that can be captured by biplanar X-ray equipment. We can swap out the foam and the mud to vary the substrate that people are walking on. People walk along this trackway. They make a footprint within that space that is captured by biplanar X-ray video. Then we take a 3D scan of the footprint that is produced as well. Using custom software, we can accurately track the 3D motions of the foot while that specific footprint was made. Then with these data, we can build 3D animations of the entire scene of the experimental trial, which includes the foot moving and deforming realistically in sync with the observed marker motions. We also register the 3D model of the footprint that was produced so we can study how the foot moved and deformed relative to it. Then we've also used new technologies to quantitatively analyze the deformation of the substrate itself. Here the credit goes to my colleague Peter. He implemented an approach using supercomputers to model the physical behavior of every individual particle within this entire volume of substrate, including the interactions with the foot and with all of the surrounding particles. With our in vivo experiments as a source for validation, we could then accurately simulate the substrate that we used in the lab. We could drive our animated foot through that virtual substrate using the motion and deformation data that we actually observed in the experimental trial. This method ends up producing simulated footprints that are exceptionally close to the real experimental ones. In this case, the average difference between real and simulated footprints was less than 2 millimeters. Most importantly, though, this simulation approach, it opened what had been a black box and allowed us to study how feet and substrates interact and to understand how biomechanical signals get recorded in footprint morphology. In other words, we can understand the relationships between form and function as pertain to hominin footprints. With the rest of our time, I want to highlight just some of our recent results which have come from our development of this new approach. And if we zoom out and think about key questions and hypotheses about the evolution of human locomotion, biological anthropologists have focused a lot of attention on our arched feet. Longitudinal arches are often thought of as one of the most important evolutionary innovations of the modern human foot. No other ape has a foot that is arched like ours. and classic papers have described this configuration as essential for allowing our foot to act as a stiff propulsive lever during bipedal locomotion. Now, that view has changed over time and become more nuanced. For example, others have suggested that perhaps arches are more important for the way that our foot stores elastic energy during endurance running. But one way or the other, our arched feet are still thought of as integral to modern human bipedalism. It's very hard to use fossil foot bones to understand much about arches, though, not only because samples of fossil foot bones are typically small and fragmentary, but also because the arched configuration of our feet has a lot to do with the anatomies of ligaments and other soft tissues that don't typically fossilize. Evidence that's long been thought of as much more reliable or more convincing for evaluating the arches of feet comes in the form of fossil footprints. Footprints like this one from Laetoli have been cited routinely as clear, indisputable evidence of arched feet in hominins going back millions of years. And that conclusion is based on some intuitive rationale. These footprints have a raised area about midway along their length that looks pretty similar to what you would see if you lifted up your own foot and looked at the bottom of it. But if we're being honest, we've never really known what this arch shape of footprints actually means. Maybe it reflects the anatomy of the foot that created it, but maybe not. Footprints are formed through this complex interaction between a deforming foot that is moving through a deforming substrate. So it's very possible that something more complicated is going on here. So we decided to investigate this question for the first time using those biplanar X-ray experiments and simulation techniques that I just explained to directly visualize and analyze how feet and substrates interact when longitudinally arched footprints are formed. What we found is that the arching of footprints seems to have more to do with overall lower limb kinematics than it does foot anatomy per se. When we walk bipedally, we strike the ground with our heel, we smoothly rotate our foot so that it's flat, and then we push off the ground with our forefoot. This motion pattern is important because it actually increases the effective length of our lower limb. We walk with what's called an inverted pendulum approach, and by rotating our foot in this fashion, the pivot point of that pendulum is actually located below the ground. This is one small piece of what helps us to be as energetically efficient as we are when we walk bipedally. So we could visualize the foot's heel-to-toe rollover in our experiments by mapping the trajectory of a pivot point that separates the parts of the foot moving up, indicated by the red vectors here, from the parts moving down, indicated by the blue vectors. And this pivot moves gradually from the heel towards the toes during the time the foot is in contact with the ground. When the foot moves in this fashion within a deformable substrate, the substrate will move down as the corresponding parts of the foot move down. And this rotational motion eventually generates a longitudinally arched footprint. As an exaggerated theoretical test, we applied this advancing pivot rotational motion to just a rigid and flat block. And through substrate simulations, we saw that even that block could produce longitudinally arched footprints as long as it was using that particular motion pattern. In other words, it seems to be the motion pattern and not necessarily anything about foot anatomy that leads to the production of longitudinally arched footprints. After making that discovery in the lab, we then came up with ways to quantitatively measure and compare the arches of footprints. This provides us with a way to comparatively analyze foot kinematics, how various feet were moving through the mud as individuals were walking and making footprints. Now, the pattern of foot kinematics that humans use generates a strong and consistent signal that gets reflected in the shapes of our arched footprints. Here I'm plotting a logarithmic fit that's based on human footprints I've collected in experiments over the past decade or so. On the y-axis is relative arch volume, or RAV. That's a normalized measurement of arch height. On the x-axis is relative depth. And so we see that human footprints tend to get more arched as they get deeper. And that's because our heel-to-toe rollover gets exaggerated when the ground can move beneath you. Now, I should mention as well that I'm plotting here footprints that were made by both habitually shod people and habitually barefoot people. And despite a reasonable range of foot morphology that's represented in this plot, humans tend to follow similar patterns. To demonstrate this further, I can even throw into the mix some 400 to 500-year-old human footprints from a site called Walvis Bay in Namibia. And we see that even these fit very well within the kinds of trends observed in our experiments. In a paper last year, we showed how the 3.66 million-year-old Laetoli footprints, with prints from these G1 and S1 trackways plotted in green here, deviate from that human trend. They're flatter than what we would expect from similarly deep human footprints. And we explained how this is a strong indication then of differences in their foot kinematics. Their feet were moving differently and they were walking differently in some way than modern humans. We further highlighted how this other Laetoli footprint from a different spatial area but from the same geological level, shown in pink on the plot, is different and much flatter than the other ones from Laetoli. This supports another paper that I worked on with colleagues where we suggested that two different patterns of hominin bipedalism are evidenced at Laetoli. And that's exciting because it opens up new ideas and questions about whether and how hominin species who lived alongside one another might have interacted, competed, and potentially influenced each other's evolution. Now, if I had a sample of these 1.5 million-year-old footprints that my colleagues and I excavated from sites near Illiret, we see that these ones follow very closely those arching patterns observed in modern humans. This tells us that these hominins had feet that moved like ours and that probably looked quite a bit like ours too. So this provides a rare piece of direct evidence supporting the hypothesis that significant changes to locomotion occurred in Homo erectus and differentiated them from their predecessors. These changes to locomotion might have been associated with a whole series of adaptations that characterized later members of the genus Homo, including Homo sapiens. So to wrap up and summarize quickly what we've found, first, there are a whole lot more fossil footprint sites out there than previously thought, so there's a lot of new and exciting data to work with. Then with new technologies and new methods, we've been able to learn new things about the evolution of human locomotion. We're gaining new insights into how extinct hominins moved. But then we're also learning new things we didn't expect. For example, our analyses have allowed us to identify locomotor diversity. That's something we couldn't do previously. In some cases, this has led to hypotheses about hominin sympatry, when there's evidence for multiple species sharing the same immediate landscape. And so these analyses have also led to insights that extend decently far beyond just locomotion. So with that, I want to thank a long list of collaborators and co-authors who were involved with all of the research I presented today. I want to thank all of my students who have been involved in all aspects of this work too. Of course, I want to thank the organizations that funded this research. Thank you again for the invitation to join you all, and thank you for listening.
If I may add a question, maybe for the audience that is not familiar with all your works, maybe you could say a few words about how I'm thinking on Illiret, of course, how footprints also give some indication about behavior in general, group composition, and things like that, and sexual dimorphism.
Right.
Okay, I don't ask you to give another talk, but in a couple of sentences.
Yeah. So there is a lot more that we can learn, because with any of these sites, when we excavate a site that has footprints like this one, there are footprints of many other individuals that exist on the same continuous surface. We know that those footprints, to be preserved on the same surface, must have been formed within a matter of hours to days of one another. If one of these sites is exposed for very long, it will develop cracks through the mud, or other animals will trample over it, and you don't get anywhere near this type of preservation that we see. And so there have been some other papers that we've worked on where we've looked at things like group behavior, group composition. If we can estimate body sizes of all the different individuals on one footprint surface, we can come up with a rough estimate of how many males, females, children might have been at that site. We can at least come up with an estimate probabilistically. We can also look at things like coordinated movement. So whether multiple, if we have 5 individuals who are all walking in a parallel direction, maybe they were oriented towards some common endpoint, they might have been walking together as well, if it just so happens that their estimated speeds match up too, which we can estimate from stride length. And then we also get a good picture of the local ecology too. Like I said, with the one new site that we're just working to publish right now, We're excavating that one, and we see all sorts of, sorry, I have to go back through all of my videos here. But we see all sorts of different animals represented here. And so there are these massive storks that were probably 6-7 feet tall that are all over the place on this footprint surface. That's an animal whose bones hardly exist in the fossil record, so this is the last appearance potentially of them by several million years. So we're gaining some great insights into animals that were on this landscape too, again, at that exact same time in the same immediate environment, sometimes capturing things that we don't necessarily know of from other parts of the fossil record.
Thank you, Kevin.
All right, thank you.